Welcome. My name is Jennifer Roeder. I'm a communications consultant for APFED, and I'll be moderating today's webinar. Today, we will learn about anti-eosinophil therapies, both where we are now and where we are going, from Dr. Calman Prussin. Dr. Prussin is the Chief Scientific Officer for Aretia Therapeutics. Prior to joining Aretia, he was an investigator within the Laboratory of Allergic Diseases at the National Institute of Allergy and Infectious Diseases at the National Institutes of Health. His research focused on T-cell pathogenesis of eosinophilic inflammation. Welcome.
Thank you, Jen. Trying to share my screen. Well, thank you all for joining us. It's a real pleasure to speak on area I've worked in for the last 25 plus years. And just some disclosures and conflict of interest before we start. I'm an employee of Aretia Therapeutics, which is developing dexpramipexole, an investigational asthma drug in which I have financial interests. I'm speaking in my capacity as an individual and subject expert. The talk contents reflects my views, but not that of Aretia Therapeutics or APFED. I'll be discussing multiple investigational drugs that have not been approved by the US FDA or other regulatory bodies. And everything that I'm going to be presenting today is in the public domain. There's nothing proprietary. So I want to acknowledge APFED, both for this opportunity to speak with you and more broadly in their work in eosinophilic disorders. I've worked with them over the years in a number of different areas, and I just think having that connection to patients and providing that support for patients is just really an amazing resource. So thank you. And also wanted to thank the patients that I've worked with over the years who've participated in our clinical research studies at NIH, as well as those of colleagues that I've worked with. Again, something that's just an amazing resource that has allowed us to make progress in these areas. This is some of the contents I'll be talking about today, and we'll drill into each one as we go forward. And just to remind you, this is National Eosinophil Awareness Week. So again, reach out to friends in that regard. So I wrote this because I think as you go through this talk, I want to underscore how much progress has been made, and that progress has been largely based on the fact that we understand these diseases, and you really can't treat what you don't understand. And I'm particularly concerned with the cuts in the NIH budget over the last few months and how the repercussions of those in terms of ultimately patient care. And I think as we go through some of the topics today, you'll understand that linkage between progress in research and progress in patient care and outcomes. So what are eosinophils? Eosinophils are a evolutionarily maintained cell type. They go back to invertebrates. You can see them in stingrays. And here you can see some examples from this fun paper by Steve Ackerman and colleagues in Journal of Clinical Allergy and Immunology, where they have photomicrographs of eosinophils from here, the tortoise, flamingo, dog, human, but all going all the way back to invertebrates. So eosinophils have been with us for millions and millions of years. The discovery of them is a little more recent. In the late 1900s, Paul Ehrlich first described eosinophils. And their association with asthma similarly goes back into the 19th century with the work of Ernst von Leyden describing the Charcot-Leyden crystal and its association with asthma. So the linkage between eosinophils and asthma goes back more than a century. And it's not surprising that therefore we tried to develop anti-eosinophil therapies as treatments for asthma fairly early in the 21st century. And the first efforts were really quite a bit of a misstep and associated with failures. You can see this top paper there, eosinophil role remains uncertain. Um, these initial studies weren't very encouraging. In the middle section, IL-5 inhibitor fails in phase 2 resistant asthma trial. And so by the middle of the first decade of the 2000s, we were really quite sort of depressed with the status of the eosinophil as a therapeutic target. And it wasn't until 2009, the work from Ian Pavord and Andy Wardlaw's group at the University of Leicester in the UK, that they really turned this around. And this work, which was here in the New England Journal, what they did is, and they didn't just look at asthma, they subtyped asthma into eosinophilic asthma, and looked at those patients with high eosinophil counts in their sputum and what the mucus that they coughed up. And in those patients who had high sputum eosinophils, or eosinophil, what we now call eosinophilic asthma, they showed an effect. And you can see here on the left-hand panel, very nicely, they could lower the blood eosinophil counts, in this case with mepolizumab, anti-IL-5. But more importantly, on the right-hand panel, where they decreased asthma exacerbations, about 50%. And that really started the current sort of revolution, if you will, in biologics in asthma and eosinophil-associated diseases, and really got things restarted after those failures of the early 2010s or 2000s. So we talk about eosinophilic asthma. No doubt if you have asthma or associated disease, you hear about T2 high asthma. I just want to go over some of the similarities and differences. So we talk about type 2 cytokines, or here type 2 effector cytokines, and there's really three: IL-4, 5, and IL-13. And these were originally characterized as being produced by Th2 cells, hence the type 2 name. But now we know they're also produced by innate lymphoid cells, basophils, and eosinophils themselves. And I want to focus on IL-5, which I like to refer to as eosinophil happy factor. It does everything an eosinophil want to make it the more of them and bigger and nastier, and they survive longer. So it's the major eosinophil active cytokine. And then the other big cytokine to think about is IL-13, and its role with eosinophils in particular is it helps eosinophils traffic to tissue where they're going to do damage. So that's the two cytokines we'll talk a bit about today. We also sometimes talk about biomarkers, and I don't want to get into details on it, but just to say that biomarkers are tests typically that we can measure to get some sense of clinical outcomes, or in this case, mechanistic pathways that are operating in patients. And so we typically measure eosinophils in blood as a biomarker for IL-5, or a readout for IL-5, and exhaled nitric oxide in patients with asthma as a readout for IL-13. And that may come, as you'll see in some of the talk. In addition to these type 2 effector cytokines, we also have these upstream cytokines. And no real details on these in terms of what they're doing, except to say that they work upstream and drive some of these effector cytokines. And the one of most note is TSLP, or thymic stromal lymphopoietin. And that's because there is a drug that targets that cytokine, tezepelumab. So what's the difference between eosinophilic asthma and T2 high asthma? We define eosinophilic asthma simply as asthma associated with elevated eosinophils, and that can be in the blood, the sputum, pretty much anywhere you measure. And then T2 high asthma is asthma associated with any one of a variety of T2 biomarkers. That could be eosinophils, could be exhaled nitric oxide, could be IgE. And so there's a lot of overlap between these two entities. So we've talked about eosinophils, but what do they do? How do they cause disease? And there's a number of different mechanisms, all of which are still being worked out. But you see here this photomicrograph on the right that the eosinophils filled with these electron-dense dark granules, which are filled with eosinophil granule proteins. And these are largely cytotoxic proteins. They're cell-killing proteins. And so you can imagine when an eosinophil releases them in the tissue, it causes tissue damage and disease. Additionally, eosinophils produce inflammatory cytokines. Cytokines are immune hormones, if you will, causing effects in other cells. And so that's another way, such as IL-13 as an example. And then lastly, eosinophils produce reactive oxygen species. You think of ozone, even it's a little more complicated than that, but oxidating reagents. And and reactive oxygen species produced by eosinophils have been shown to play a major role in the generation of mucus plugs in asthma. And so thereby causing more severe asthma. So to date, we have 6 approved biologics in the eosinophil allergy space. The first was omalizumab, anti-IgE. And you can see, as I mentioned, there's this huge gap between when that was approved and when the anti-IL-5s, because we were really figuring out how these drugs work and in what patients they work. But in short order, we've seen these anti-IL-5, anti-IL-5 receptor drugs, all of which lower eosinophils, and that's the main way they work. So I call them focused anti-eosinophil drugs. And then dupilumab and tezepelumab, which have broader T2 activity. They're not as focused just on eosinophils, even though they do have some eosinophil activity as well. So how have these biologics informed more effective therapy? I mentioned before, if you don't understand something, you can't treat it. And certainly we learned that lesson with the anti-IL-5s. We didn't understand the disease well enough and how these drugs worked in disease. But once we learned that they work in eosinophilic asthma or T2 high asthma, that really became a success. Very similarly, we've learned from the failure of these same drugs, these focused anti-eosinophil biologics in eosinophilic esophagitis. So, for example, benralizumab in this study in the New England Journal from last year, it does a beautiful job lowering eosinophils in tissue. You can see that on the leftmost panel. So it works. It lowers eosinophils just like it's supposed to. But as you can see in panel B, it doesn't do much for symptom scores in these patients. So it makes the eosinophils go away, but not the disease symptoms go away. And so that's really taught us that these very narrowly focused anti-eosinophil drugs may not be adequate in some diseases. Conversely, dupilumab, which is an anti-IL-4 receptor alpha drug, a bit broader anti-T2 focus, has shown efficacy in eosinophilic esophagitis, and in fact is now an FDA-approved drug for that disease. And on the right-hand panel, you can see a nice effect of the drug and separation from the placebo. Another lesson we've learned, what are the consequences of depleting eosinophils? And you can see these 3 papers that basically address the same question. What happens when you get rid of these evolutionarily conserved cells? And so far, the answer in humans is not much. Now, these drugs, it's still early days. They've only been approved. The first one is just getting on 10 years. But thus far, we haven't seen a real safety signal or downside of that. And so obviously, there's something that people will continue to monitor for and learn about as these drugs become increasingly used. But thus far, they appear to be quite safe. I also always like to share a picture from my garden. So this is my Chinese windmill palm, Trachycarpus fortunii, and it's about 25 feet tall. I started out, it was a little seedling, and it survived this last winter where we got down into single digits in Maryland. So I'm just thankful that it's still around. So just always like to share that. So where are these drugs going over the next decade? Um, and I think there's four general directions that I'd like to address in this. Um, so the first, all just talking about these biologics. Um, Most of them are antibodies, and this is a schematic of an antibody. You can see there's two ends of it. The FAB end is the antigen binding domain. So that's the part of the drug that binds the antigen, like your hands grabbing onto something. And then the back end, the FC domain, is a functional domain. It provides function to that antibody. And the best example I can give is with benralizumab. So benralizumab has been altered so that it lacks the fucose, which is a sugar, on its FC domain. And by not having fucose, that increases target cell killing. So benralizumab kills eosinophils because it has that, it's afucosylated. It has very essentially no fucose on it. In a very different way, you can make modifications to the protein, um, antibody protein. In this case, way you can make it what's called a YTE modification in that FC, and that increases the serum half-life of the antibody. So the antibody sticks around longer in the serum, and so you have to dose it less frequently. And the best example of that in the eosinophil space is depemokimab. So that's a long-acting anti-IL-5. It's a humanized anti-IL-5 molecule that's engineered to provide an extended half-life, and it's higher affinity. It grabs on tighter to IL-5 compared to the approved anti-IL-5s like mepolizumab. And you can see an example of that here. This is a early phase 1 trial. And what we're looking at here, so on the y-axis is the eosinophil count. And you can see in that black line at the top where my cursor is, that's the placebo. Doesn't really change. It's just sort of a straight line. And these others are all these different doses. And what's important is the highest doses here, the purple and gold lines, were able to stay within that target range, about 80% reduction, through 26 weeks. So that's through 6 months. They provided adequate eosinophil lowering, at least what they think would be clinically effective. So then you go and do a clinical trial, and indeed, these phase 3 trials were done and read out last year in the New England Journal, and in the SWIFT 1 and 2 trials of depemokimab, and they gave 2 doses of the drug at the beginning of the trial at week 0, and then again at week 26, so it's twice weekly. And they saw about a 50% reduction in severe exacerbations. And you can see that the separation of the placebo and the depemokimab. And these are just the cumulative incidence of exacerbations. So the next drug I'd like to talk about is dexpramipexole, and that's the drug I've been working on for the last 9.5 years. It's an oral eosinophil lowering drug. It's a small molecule drug. It's not a biologic. So again, you take it orally. And it works by inhibiting eosinophil maturation. And you can see this cartoon of eosinophil maturation through these various stages. And it interrupts that between the eosinophilic promyelocyte and myelocyte stage. And we've seen evidence of that both in bone marrow biopsies taken from patients as well as in vitro culture systems. Here is the phase 2 proof of concept study we performed. On the left-hand panel, you can see the different dose groups. So it was about 100 subjects, patients, and participants total. And you can see in 3 doses of drug and placebo. And you can see in the highest dose, we got an 80%, approximately 80% reduction in the blood eosinophil count. And on the right-hand panel, you can see the same data, but shown for all the different time points of the study. So you can see that it takes a few weeks for the drug to start to lower eosinophils. It reaches a maximal eosinophil lowering at 12 weeks. At that point, we stop the drug, and you can see almost the mirror image in terms of recovery of eosinophils. So here, this study was a small phase 2 study, proof of concept. It was not powered to look at lung function, but of course we did look at it. And here you can see FEV1, the amount of air you can exhale out of your lungs in 1 second. And you can see there's a substantial increase in FEV1 in the dexpramipexole 150 BID, so that's the high dose group, relative to the placebo group. You can see that week 8, week 12. And what's interesting is at week 16, 18, so this is 4 to 6 weeks after stopping drug, we still saw a substantial difference relative to placebo. And that's because the drug, after you stop it, it takes several weeks for the eosinophils to come back. And so even off drug for a few days here or there, patients still, their eosinophils are controlled. So the next topic area I want to talk about was bispecific antibodies. So these are monoclonal antibodies that block 2 or more targets. So we talk about bispecific, trispecific, et cetera. They're generated using a variety of bioengineering approaches. They allow for the inhibition of multiple inflammatory targets and pathways with a single drug. Thus far, most of the development has been in cancer, but they're rapidly expanding into immunologic and inflammatory diseases. And I suspect they're going to be one of the next big things to hit the clinic. So the example I picked for this was lunsekimig, which again is a pretty unique molecule. It's composed of what are termed nanobody domains. So these are individual antibody domains. They're actually originally developed in camels, and so they're sometimes termed camelid antibodies. But again, all through genetic engineering, these are pasted together, if you will, hooked up together. And so in this case, you have 2 domains blocking IL-13, 2 domains blocking TSLP, and then a 5th domain that binds to albumin. Albumin's a protein in the serum, and by binding albumin, the drug hangs out more in the serum and doesn't get catabolized and degraded. And so it blocks, but this drug, since it blocks IL-13 and TSLP, it's blocking both upstream TSLP as well as an effector Th2 cytokine. So a very different take from the currently approved drugs in terms of combining both of those approaches. So in this phase 1b proof of concept study that was published this year, a single dose of lunsekimig or placebo was given, and they looked at various biomarkers. In this case, exhaled nitric oxide on the left, and you can see a very nice separation of the lunsekimig versus the placebo groups, and about a 50% reduction in exhaled nitric oxide. On the right-hand side, you can see blood eosinophil count, and again, a nice separation of the drug treated from the placebo, showing a decrease in blood eosinophil count. Again, this was a small clinical trial, so 36 participants total. And so it was not powered to look at lung function. That requires multiple hundreds of patients. But you can see here, looking at FEV1, that you see a nice separation of the drug-treated patients from the placebo in terms an improvement in FEV1 in those drug-treated patients relative to placebo. So certainly encouraging in that regard. And then the last category of drugs I wanted to discuss were chimeric protein degraders. So there's lots of different names these go by: protein degraders, heterobifunctional degraders, proteolysis targeting chimeras, or PROTACs. These, again, have initial development was in cancer, but now there are multiple development programs and multiple companies developing these in immunologic and inflammatory diseases. And these are another small molecule pathway, so an oral drug to target these specific pathogenic pathways. And it's very early days, particularly in regard to inflammatory diseases, but certainly there's a reason that to believe that these will make it to the clinic eventually. And the way these work is really quite interesting. So what they do, this drug has, just as it said, it's heterobifunctional, as two ends. One end binds the target disease-causing protein that you're trying to get a hold of, and the other binds an E3 ligase. And when you bind them together, what happens is you take this disease-causing protein and it gets shuttled into the proteasome. And the proteasome can be thought of as the cellular trash can. So it takes this disease-causing protein, shoves it in the trash, and gets rid of it. So it's a way to target drugs very differently from monoclonal antibodies, but with that same specificity. So the one that I'll be talking about is targeted at STAT6. So what is STAT6? So STAT6 is downstream of interleukin-4 and interleukin-13, very simply. So in order for interleukin-4, IL-4, and IL-13 to work, they need STAT6. And you can see an example of that on the right. So this is a mouse asthma model from 25 years ago, but still just as good as ever. On the left-hand side, they performed this asthma model in a wild type, a normal mouse that has STAT6. And you can see all throughout these areas of mucus hypersecretion and hypertrophy, where these purple mucus cells are staining very differently from everything else. And when they knocked STAT6 out in these mice, so these mice have a genetic deletion in STAT6, on the right-hand side, you can see there's no hypersecretion of mucus, that the bronchi in these mice, the airways in the mice, essentially look normal. So the drug we'll be talking about, KT-621, is a STAT6 degrader. It has just started human clinical trials, so phase 1, early phase clinical trials this year. But there's preclinical work, in vitro laboratory work that's been done on this that I'll share with you. So what you're looking at on the left is, again, in vitro work using different types of cells that occur in human airways. And we're looking at, on the y-axis, is the amount of STAT6 in these cells, so just percent. And then on the x-axis, we're increasing the concentration of this drug in these cultures. And you can see a very nice dose response. As we increase the drug in these cultures, we can turn down the amount of STAT6 and pretty much get rid of almost all of it. So that looks good. Can get rid of it in these tissue cultures. On the right-hand side is showing the specificity of this drug, that it actually hits STAT6, but nothing else. And so what's being done here is using human blood cells, peripheral blood mononuclear cells, treated in vitro with the highest dose of this KT-621 that we're seeing on the left. So using these high doses of the drug, we're performing and treating these human cells in vitro. We're then performing, or we, is not me, so I shouldn't say we, they, the group that did this, perform proteomics, where they're looking at all the proteins in these cells and saying what amounts, what has changed between after we put in the STAT6 degrader versus just putting in a solvent. And we're looking at a volcano plot. This is a bioinformatic plot where the x-axis shows fold change and the y-axis shows the significance, the p-value. And you can see where my arrow, only one protein pulls out as significantly changed, and that's STAT6. And all the other proteins in the cell have not been changed. And more importantly, all these other stats, which you might say, well, maybe it would mistake one stat for another. All those other stats also were not changed. So it really looks quite specific. And that's reassuring that this drug is not going to have potentially any off-target effects, that it's really going to be limited to STAT6. And thus far, all of our understanding of STAT6 is it's limited just to this IL-4 and 13 system, and it's not playing a role anywhere else. And obviously that's going to be very important moving forward. So that's working in a test tube, if you will, in vitro. And then here we have the same STAT6 grader in a mouse model of asthma. So this is a house dust mite allergen asthma model, where mice are exposed to house dust several times a week, which generates an eosinophilic response in the lungs. And then they're treated either with the STAT6 degrader, KT-621, or treated with dupilumab, the approved anti-IL-4 receptor alpha drug. And so you can see in this middle panel where we're looking at lung eosinophils and the bronchoalveolar lavage from these mice. So first of all, where my cursor is in a mouse that hasn't received house dust mite, there are no eosinophils in their lung. In the next over, you can see when they're just given vehicle, so they're not given the STAT6 degrader, you get this whopping eosinophil signal with this big black bar. And then in the mice that were treated either with the STAT6 degrader or with dupilumab, the IL-4 receptor blocker that blocks IL-4 and IL-13, either of them pretty much blocked eosinophils close to 100%. So really very convincing that, that this STAT6 degrader works in this mouse model of asthma. Moving over to the right, in the same model, looking at goblet cell metaplasia. So these are changes in the mucus-producing cells along the lines of what I showed you a few slides back in that airway, this increase in these mucin and in the size and number of these mucus-secreting cells. And again, you see this big black bar, this big whopping signal in the house dust mite model. And for both the STAT6 degrader as well as for dupilumab, a significant reduction in the mucus production. And mucus is a major player in asthma. So these are encouraging preclinical data, and we'll see over the next few years how this drug plays out in the clinical space in asthma, atopic dermatitis, and possibly other eosinophilic diseases. I want to end just on a note that I'm very optimistic, particularly compared to where we were 10 years ago, where we were really struggling to figure out a path forward. We've got a much better understanding of the biology of eosinophilic diseases, eosinophilic inflammation, asthma, and atopic dermatitis, eosinophilic GI disease. What are the driving forces? And have much better tools to try to treat them. And I can only imagine that things will continue to get better as we go forward. So pleasure talking with you, and I'll take questions at this point.
Thank you so much for such a wonderful presentation, going into so much detail about all the potential therapies and the therapies that are already available. So as Dr. Prussin referenced, we're now ready for questions. So please click on the Q&A tool at the bottom of your screen, and you can type in some questions. We're going to have a discussion here. So the first question we're getting is about LDN low dose naltrexone for EOE. What information are you seeing about this therapy? This person has heard positive feedback and curious if that's what you're seeing as well.
Yeah, so that really is not an area, not an approved drug for these diseases, and I have not seen published investigations of naltrexone, and so I can't really comment on that.
Great, thank you. Have you seen published information about Jorveza, the dissolvable tablet that's available in other countries? People are curious about what the status is in the United States.
Yeah, so that that's really a regulatory point of view. I think we've all so this is yeah. So it's really it's not a scientific point of view. It's really a regulatory discussion that's ongoing.
Okay. So let's talk a little bit broader then. For your point of view, what are the challenges in developing therapies for eosinophilic diseases?
So one of the big challenges is really, how do you measure patients are improving? How do you measure patients are better? So for example, in hypereosinophilic syndrome, when you do a clinical trial, you have to be able to measure something and say, not just I lowered eosinophils, but I made somebody's life better. I made made them feel better. And although you think that would be a pretty easy thing to do, it's actually quite difficult. And so the development of outcome measures, you saw in some of my slides on EOE, that the dysphagia symptom patient-reported outcomes have really been advanced over the last 5 to 10 years. And so though when you have one of these patient-reported outcomes, and there's a whole process of how you develop it and validate it, that gives you something that you can measure, which you can then take to regulators and say, look, I've got a validated outcome. I can show somebody's actually improved. We've worked with Panise Khoury at the National Institutes of Health, or I have worked on a patient-reported outcome for hypereosinophilic syndrome. Because again, that's been a real struggle. What are if you want to get a drug approved, you have to be able to measure something and show it's improved. Just showing eosinophils are gone is not enough. As you saw very clearly from that benralizumab study, you actually have to say what's clinically improved in a patient.
Thank you. As we talk about a variety of different conditions, one of the topics you referenced was different biologics. How are emerging biologics different from existing treatments, and kind of what gaps are they trying to fill?
Yeah, so I think what I showed you is more or less what it is. So one is dosing. So for example, dupilumab is dosed every 2 weeks. And having and having something that's dosed every 6 months versus every 2 weeks, obviously patients like that. And so longer dosing intervals, you're seeing a lot of energy in that area as in terms of clinical development of new drugs. And then the other is, again, like I showed, the bispecific. So for example, there's a number of patients with hypereosinophilic syndrome where they're treated with an anti-eosinophil drug, but may also get one of these second upstream anti-T2 drugs like dupilumab or tezepelumab. Now, obviously, that's easier said than done because one, they're very expensive, and two, there's not a lot of information on safety of the two together, and we obviously want to be really careful in that regard. but developing new drugs where the two things are combined from the start, so we understand the safety from the start, has the potential to have a broader effect on inflammation, on disease mechanisms.
Well, touching off that, we've got a follow-up question that came in around biologics and how you talked about combinations. So, what kind of research are you seeing about combinations of biologics for conditions like EOS
Yeah, and
asthma,
that's
EOE?
yeah, and that's a hard one because on there have been for example, there was a paper published uh, in the New England Journal a couple years back. Mike Wexler is the first author in asthma, looking at combining dupilumab, which blocks IL-4 receptor, okay, and then also adding that an anti-IL-33, and combining those and see do you get improved efficacy. The difficulty is severalfold. One, drug companies tend not to play well outside of their own sandbox, if you will. And so if you have one company with one drug and another with another drug, how do you combine them? So that's been just structurally an issue um, in terms of doing studies combining some of these currently approved drugs. And I think that's one of the reasons why going forward with these bispecific drugs, where the original product has that combination, it gets around some of those structural impediments. Um, Over time, uh, institutions such as the NIH have done these combination studies. And so it's not that they don't happen, but they don't happen as quickly, unfortunately, just due to some of these structural artifacts and impediments, because we'd all like to see some of that, but it's how do you actually actually operationalize that? That's more difficult.
Thank you. Could you speak at all about TEDspire and its potential?
So Tespire works upstream. And so it works upstream of IL-4, IL-5, and IL-13. So I think it has a very good potential for a number of uh, eosinophilic inflammatory diseases, such as eosinophilic esophagitis, eosinophilic gastritis, duodenitis, and such. Um, but ultimately, it's you do the experiment. This is all why we do science. It's because we can think all we want about these things, and until we actually get data and evidence, it's just thinking. But no, I'm very encouraged that that tezepelumab, Tespire could uh, be an ex, be have efficacy in some of these eosinophilic inflammatory disorders.
Thank you. Are any of the upcoming potential new therapies looking to target eosinophilic fasciitis?
So I am not aware of what is being done in terms of actual clinical development from other companies. So I can't really speak to whether it's going to be through a company or through some consortium of investigators. I expect that, given the success of dupilumab in eosinophilic esophagitis, that would be an obvious next step, but I can't speak to that in terms of actual boots on the ground clinical development.
Thank you. We've got another question about a potential future one. They're curious, are you familiar with ultrasound guided intro-lymphatic injections that are used in some asthma clinics and oncology trials? Do you see any potential crossover for other drugs to be delivered in this way for eosinophilic conditions?
So I'm not familiar with that technology, but um, I can tell you that, you know, always, the KISS principle, keep it simple, stupid. And unless you have some amazing technology and clinical effect, doing an intralymphatic injection and the amount of expertise. So unless it's going to put somebody into, one treatment and you're done, I have, it doesn't, I have concerns that it's again, and I know very little about that specific. But when I've seen other approaches like that, it's a lot of work. And that's one of the reasons why there's so much of a move to oral medications or attempts to move to oral medications, because patients like simplicity, and intralymphatic injections are about as uh, complicated as I can imagine. So yeah, I um, I think there's so many other solutions coming down the line that to me that just seems very complicated.
Thank you. So you talked a bit about potential treatments and existing treatments for eosinophilic asthma. Is there overlap with treatments being used to target neutrophilic asthma?
Yeah, that's a hard one. So eosinophilic asthma is a very clear entity, and it's not that, and we understand it, and we understand what sort of turns it on and what drives it. We understand, what drugs treat it and what, all those things. And we just don't have that with other types of subsets of asthma. So you could say, well, yeah, you do. You understand that, IL-8, it has a role in neutrophilic asthma, but it's not, it's a decade or more behind. And so I certainly hope in the future there will be more advances, but right now these non-eosinophilic asthmas, we just don't understand them as well, and so they're more in their infancy. That said, you look at the progress over the last 10 years in eosinophilic asthma, and it's only a matter of a few observations that suddenly things become much clearer and you can make that kind of progress. So not to say there won't be progress, but just to say we're not there at this point.
Okay. Let's switch for a few minutes and talk about clinical trials. How can patients and families get involved in clinical trials, and what should they consider before enrolling?
So in terms of finding clinical trials, I imagine, I'm probably not the best person how to find them. Although I imagine the APFED website has a listing of clinical trials. Um,
You are
clinical
correct.
trial, I'm sorry?
You are correct. There is a
Yeah.
listing on APFED's website.
Clinicaltrials.gov is an encyclopedic listing, but the difficulty is finding it. The NIH Clinical Center webpage has a page for, NIH sponsored trials. Um, but I think what I can say is two things. One, um, the way we make progress in medicine is through clinical trials. And they don't all have to be drug trials. They can be trials where patients simply come in, they have a workup and maybe give a blood sample or things, such as that, or in asthma, they have some pulmonary function tests to understand the mechanism of disease. We used to do a number of those types of trials when I was at NIH. And that can be a huge source of research material and understanding to move things forward. The other thing to remember is clinical trials, we don't know what the answer will be. And so um, you have to go into it understanding that if it's a drug trial, that you may get placebo if it's a blinded trial. And even if you're not getting placebo, you don't know whether this drug will make you better. And so, not to be a Debbie Downer on it, but the flip side is just to be sober about that realization, and to go in all eyes open. Clinical research is not for everyone as a participant because of all those complexities. And again, I think the world of people who have participated in the clinical trials I ran. They really gave something that nobody else could, and gave of themselves in a way that really was just incredible. But it's not for, I have also had patients who clearly should not have been on that trial and just joined me, and perhaps not knowing themselves well enough, and then realizing that was a mistake. So that's another, it's sort of the a negative of it, but it is, I think they can be amazing, but they can also, at the flip side is, there's a lot of unknowns, and some people do better with unknowns than others.
Thank you. Can you talk a little bit about how side effects are identified during clinical trials or after?
No, that's a huge part of clinical trials. So there's a whole structured recording system in terms of of how you record side effects, and how you name them, um, and and make them available to regulators such as the FDA. So they're, as much as possible, comparing apples to apples and understanding both the severity of the side effects, but actually what, organ system they're in. So there's several different systems. One called MEDRA, which is the medical dictionary for research something or other. But MEDRA's used quite extensively. And then there's a second that the NIH sponsored, CTCAE, clinical trial, uh, something for adverse events. But basically, they're very carefully scrutinized. In fact, almost all the trials I showed you, except, well, I shouldn't say, so the phase 1 trials that I showed you, their primary endpoint is safety. Safety is really paramount in all these things, and there's an incredible amount of transparency in that regard, in terms of recording of these, making them available, and to the regulators.
How long does it generally take to identify side effects?
That's a hard, when you say how long does it take, meaning, I'm not sure, in a
The
single
individual
patient or with a drug?
the individual's asking about with a drug, like how long does it generally take to find side effects in drugs?
Oh, okay, that's a good question. So it can be very quick.
Mhm.
So if it's a common side effect, or if it's a spectacular side effect, it's pretty quick. Now, when you first do a clinical study, so you do what we call first in human, and actually there's a really very cool um, television series called First in Human that was on public television, and it was done basically looking at first in human clinical trials that were done at the NIH. So there's a whole structure built
Mhm.
around when you first take a drug and put it into a human being, how much data you need from animal models to show that the drug is safe, and then how do you calculate the actual dose. And again, there's a whole what's called the International Council on Harmonization, but a global agreement that different regulatory authorities have on how these are done. And so these, you can, if you Google ICH and first in human or something like that, you can pick up the guidance. It's freely available. But exactly that. So you have to start with a very low dose, and you give a single dose. So that's called a SAD study, single ascending dose. And then you do that, and then you look at that single dose as you went up on the dose. Were there any side effects? And you're also giving placebo at the same time. And if that looks good, then you can go into what's called a multiple ascending dose, or MAD study, where you can give multiple doses and go up on those doses. Um, so you're looking all throughout that at safety and tolerability. And then you go into these larger phase 2 studies of few hundred people or 100 people, and you continue to look. You're meeting with regulators all the time during, after each one of these type stages of trials and they're going to look at your data. So the FDA gets all the raw data. They don't just get what you tell them. They get the actual raw data in terms of adverse events, patient level data. Um, and then you go into phase 3. But the difficulty is, and I think is even when you do a study in a few thousand people, if there's some very rare event, you might not see it. And so that's why we then post-marketing still um, require following adverse events in one way or the other. Now, that can be as simple as the FDA's voluntary reporting system, or if if the FDA has some concerns about a drug, they actually have a formal post-marketing study for side effects. So uh, everyone takes it seriously because we're interested in safety and efficacy, and these drugs have to be safe. No drug, though, I will be the first to say, nothing, you know, nothing is 100% safe that you do. Every intervention has some downside. It may be very rare, it may be very mild, but no intervention is completely devoid of side effects. So that's just the reality of life. And so it's always looking at benefit to risk and saying, what's the benefit of this drug? Because if you have a disease, there's also the risk of the disease. So what's the benefit of the drug versus the risk of the drug relative to untreated disease?
Thank you. So as you look to the future and consider future therapies to be developed, do you anticipate that they'll be disease modifying, or will they primarily be designed to manage symptoms and inflammation?
No, thank you. That's whoever asked that, great question. Um, and I think that is spot on, that we are in early days, and that is exactly what you want to be doing. You don't want, first of all, are there ways with whether it's lifestyle or other things, avoid these diseases? We certainly, some of these diseases have increased because we're more aware of them, but there are also clearly greater frequencies of immunologic diseases. Understanding that better, which requires research, not just talking off the top of our head, but understanding why there's an increased prevalence in various immunologic and inflammatory diseases. And there's lots of research that's been done on that and more that's needed. And can we find ways to find out what some of those culprits? I don't think it's a single culprit. It's probably what we call multifactorial, many things, but can we actually decrease some of the risk? An example of that is with food allergy. We mistakenly, as a medical community, thought, oh, let's avoid early life food allergens and we're going to protect kids. And we actually ended up kids by not getting exposed to food allergens early. Many of these kids we increased the prevalence of food allergies. So there are some modifications that we can do, and hopefully more in the future. But then going to that point, disease modifying, actually not just getting rid of, and I don't think these aren't symptoms. I mean, when you get rid of eosinophils, I think that's not just a symptom. That's actually getting rid of a huge component of the inflammation that's causing tissue destruction. One of the things which has not been done yet, but as these drugs become more available, hopefully less expensive, we can start moving earlier in the treat and shown to be safe for long periods of time. We can start moving earlier in the treatment paradigm. So not just right now, what we do is we treat people once they have pretty severe asthma and they have a lot of damage to their lungs. But treating them 10, 20 years earlier when they're early in their disease. That's what we do with cardiovascular disease. We treat people when they have high cholesterol and high blood lipids. We don't wait till they've had a heart attack, and only then we start giving them, various cholesterol lowering treatments. So in the same thing, if we can change the course of disease by starting earlier, or perhaps figuring out other approaches to modify the disease and hopefully change that course, because people with asthma lose lung function over time, and interrupting that loss of lung function. Same thing with eosinophilic esophagitis. Patients with EOE get strictures. And we want to treat these patients before they already have strictures and the damage is done. I think everyone who's, you've seen patients who have had EOE, for, 20, 30 years, and you're never going to get them back to where they were before they had that. But if we can start early and interrupt that fibrosis. And so I think that in many ways, thinking about how to modify the disease and how to interrupt the disease is spot on.
Thank you very much. Appreciate all of that information. Do you have any final parting thoughts that you'd like to share with the group about the future or current status of therapies?
No, just I wish I were 30 years younger and starting out in many ways, in some ways not. But the translation of the Human Genome Project into understanding and bioinformatics, our understanding of human biology is fascinating and wonderful in and of itself. But more importantly, I think we're going to see over the next generation that translation into human health. And so I think from that point of view, it's an amazing time to be in medicine. And I'm hopeful that that ultimately translates into better quality of life for our patients. So I'm very optimistic, and particularly in terms of eosinophilic GI disease. When I was NIH, I had a large adult eosinophilic GI disease practice clinic, and patients with particularly eosinophilic gastritis and gastroenteritis, and those are very tough diseases. And seeing some of the breakthroughs over the last few years, and I am very optimistic that those patients will continue to get better therapy as time goes on.
Well, thank you. We appreciate all of your time today to be able to share information with us. And we also want to make sure to take time to thank our education partners for their support for this webinar series. That includes AstraZeneca, Bristol Myers Squibb, GSK, Sanofi, Regeneron, and Takeda. As Dr. Prussin shared at the beginning of the webinar, this week is National Eosinophil Awareness Week. So we hope in addition to attending today's webinar, you'll visit apfed.org/NEAW to check out ways to be able to get involved, not just this week, but all throughout the year. This year, 2 additional awareness days overlap with Awareness Week. Sunday, May 18th, was World Eosinophilic Diseases Day, and tomorrow is the first ever World EOE Day. So from tomorrow on forward, May 26th will be World EOE Day. So check out information about both of those on APFED's website to learn about ways how to get involved. Just so many opportunities for awareness right now. If you'd like to continue learning more about eosinophil-associated diseases, I encourage you to subscribe and listen to our podcast, Real Talk Eosinophilic Diseases. In the latest episode, we talked with Dr. Melanie Ruffner about comparing pediatric and adult EOE. You can find the episode at apfed.org/podcast, as well as on Apple Podcasts, Spotify, Audible, and more. We also currently have registration open for our annual patient education conference, EOS Connection. This will be APFED's 23rd patient education conference for eosinophilic-associated diseases. This year, we're headed to Raleigh-Durham, North Carolina from June 26th through June 28th. But even if you can't join us in North Carolina, this continues to be a hybrid event. So we encourage you to visit apfed.org/conference and register to either join us in person or virtually. Finally, we encourage you to continue the discussion started today in our online community on the Inspire Network. It's a great place to connect with patients and caregivers for support. You can check it out at apfed.org/connections. Thank you again to everyone that joined us today for the webinar and to Dr. Prussin for his presentation. If you have a question that didn't get answered today, please email mail@apfed.org, and we encourage you to visit apfed.org/webinars to see recordings from past webinars and stay tuned for the schedule of upcoming webinars. I hope everyone has a great afternoon.